Now you've had a look at some of the terminology around genetics, we're going to have a look at an actual genetic cross. We're going to look at something called a monohybrid cross, and we'll look at the definition of that in the next slide. So I've got here, geneticists can investigate patterns of inheritance in plants and animals by studying one characteristic at a time. And that's the basis of our monohybrid cross. Crosses are set up by true breeding. Now, by true breeding, what we mean is that the individual is homozygous for the characteristic. So, for example, looking at smooth and wrinkled seeds, then smooth is the dominant, wrinkled is recessive. So you'd be looking at a true breeding individual would have the two dominant characteristics or have the two recessive characteristics. The offspring that are produced as a result of crossing these that are observed, and the different numbers of each phenotype are counted. Now, what I mean by crossing them, or a cross, is when fertilisation is taking place. So you've either got some sort of form of sexual reproduction taking place between plants or between animals. And then the offspring are looked at, and what the phenotype is counted, and that information can be used to work out whether an allele is dominant or whether it's recessive. Of a monohybrid cross is a genetic cross between two organisms that differ in only one way. And the example here is, well, I've got written here sooth versus wrinkled pea seeds, but what I mean is smooth versus wrinkled pea seeds. So here we're going to have a look at a wee example of a monohybrid cross and a worked example. Now this is looking at pea plants, and we're going to look, first of all, here are our parents. So the parents are often represented by the letter P, and the phenotype of the parents, in this case here, one of them is a tall plant and one of them is a dwarf plant. Now we know that when we're doing this cross, the parents are starting off as true breeding or homozygous. So we know therefore the genotype of the tall one will be capital T, and the dwarf one will be little t. Now these are just ordinary, if you were to look into the ordinary cells of your pea plant, they would have these genes within them depending on which parent we're talking about. But when we're looking at the gametes, that is when we're looking at the pollen that's there, or we're looking at the eggs that are there, then they have half the chromosome number. And so what is going to happen is that this is going to be halved, and the gametes can only have a large T in them, in this case over here, or a small t in them, in this case over here. So every pollen that is produced has only got a large T in it if we think of this as the male gamete. And if this is a female one, then every egg has only got a little t within it. So therefore, when these two gametes come together, in other words, your large T and your little t come together, then in fertilization, then you end up with an individual that, or the plant has got this here, a large T and a little t. Now they have done this here on a Punnett square, and I'm not going to worry about the Punnett square here on this part here. But what I am going to tell you is we call the offspring from this the F1, and the F1 that are produced in this will all have the phenotype tall, because they've all got the dominant allele here. So these here are the plants that result from this cross that is up above here. Now, I said this is known as the F1 generation. Now what happens is in an F1 generation that you then take that F1 generation, sorry, what happens in a monohybrid cross, you then take that F1 generation that's been produced, this here, and you cross them with another individual that is the same genotype, that is large T, little t. And we're going to move on and have a look and see what. So here, up here, we've got the F1 generation, and we're going to self-cross them. So we take individuals that are tall and that have the genotype large T, large T, little t, and we're going to cross them. Now, the possibility in the gametes, when the gametes are formed, so imagine you've got lots of cells over here, and they are formed into gamete cells, so the number of chromosomes is halved. So we're thinking of pollen here, and we get lots of pollen grains that are produced. Well, some of them will have the large T in them, some of them will have the small t in them. And in the same way with the eggs, some of them will have a large T and some of them will have a small t. So there are more possibilities of what can be produced in the offspring with this. Because what can happen is, well, it might be that a pollen grain that's a large T meets up with a large T in the fertilization process, or it might be that the large T meets up with a small t. So, or it might be that a little t meets up with a large T, or a little t mixed up with a small t. Now, it's kind of complicated to sort of work your cross out that way. So what we do is we use something here called a Punnett square. And in a Punnett square, what happens is the gametes for the female and for the male are put up at the top here on the Punnett square. So here's a possibility of the female gamete. It could be a large T, or it could be possible that it's a small t. And here are the male gametes. It might be possible that it's a large T, or it might be possible that it's a small t. So what you do then is you then imagine that fertilization is taking place. Now, fertilization is a random process. So it might be that a large T pollen grain might meet up with a large T female egg, and therefore you end up with this. Or on the other hand, it might be that if we work along here, a large T meets up with a small t here, and therefore you get an individual with that. Or looking here, if we move along the Punnett square here, we've got a large T up here, if we take it down to there, and a small t down here, and we put them together and we get a large T, small t. And it's just good convention just to put the capital letter first. Or if we end up where we take this gamete here, might meet up with this gamete here, and we end up with an individual who's got a small t, small t. If we have a look at the genotype ratio that is produced from this cross, or this generation, which is known as the F2 generation, if we have a look at the genotype ratio, then we discover that we've got one large T, we've got two large T, small t's, and one small t, small t. If on the other hand we have a look at the phenotypes of these, so the phenotype ratio, we find that this here is a tall individual, this also is a tall individual, and this one here is a tall individual. So we've got 3 tall, and this one here is a dwarf individual. 3 tall to 1 dwarf, and that's a standard monohybrid F2 generational cross where you get a 3:1 phenotype ratio. Now, I've got here a predicted versus the reality. The thing is that I've said that you get this expected ratio of 3:1. in actual fact, in reality, the expected numbers can differ from the actual numbers, and that is because fertilisation is a random process and it's got an element of chance. So you don't always get the 3-to-1 ratio exactly. When Mendel did his experiments, he did them with huge numbers of plants, and so the more crosses that you do, the more likely you are to get that actual number. But that is the basic monohybrid cross, and we'll go on to look at examples of them and try and work our way through to understand this when we're in class.